Good afternoon, everyone, and welcome to the 2024 Lister Family Engaged Science 3-Minute Thesis Competition. My name is Ingrid Shiraz. I'm the Project Administrator for the Office of Student Academic Services here at the Faculty of Agricultural and Environmental Sciences. Today's event is supported by the Lister Family Engaged Science Initiative, which provides science communication training to Macdonald Campus students. It also provides different opportunities for our students to apply the different skills they've developed, such as today's competition. We would like to extend our heartfelt gratitude to Mrs. Lister and her family for their unwavering support to the initiative and for their generous contribution to today's competition. We are also proud to announce that we have received the gold certification for the McGill Sustainable Events Program, which is run by the Office of Sustainability. I'd like to start the event with a land acknowledgement. The Ganinge-Haga Nation are the custodians of Tiohtià:ke, the unceded territory on which McGill University stands. They're also the custodians of the surrounding lands and rivers. To the north resides the communities of Kanesatake. To the south, the communities of Kahnawake. We would like to thank them for having watched over these lands for so long and for continuing to watch over them today. This year, we have 9 brave Macdonald Campus graduate students that have taken on the challenge of presenting their research in 3 minutes with only 1 static slide to a very diverse audience. We're very happy to offer our students an additional prize. In collaboration with graduate and postdoctoral studies, our first place winner will automatically qualify for the final of the McGill-wide 3-minute thesis competition taking place this spring. So a big thank you to their team for providing this wonderful opportunity to our students. We truly appreciate it. Our judges, I will switch. Are going to determine the winners of first, second, and third place, and you, the audience, both in person and online, will get to vote for your favorite talk and determine the winner for People's Choice. Our judges this afternoon are Dr. Jean-Benoît Charon, Associate Dean, Graduate Education at the Faculty of Agricultural and Environmental Sciences, Dr. Catherine Panucco, Professor at the Department of Biology at John Abbott College, and Mehtab Singh, PhD candidate in plant science and the first place winner of our 2023 competition. Now, please welcome our event moderator, Dr. Alice Shirestis, Associate Dean Academic and Director of the Office of Student Academic Services.
Thank you very much, everyone. Thank you for attending, and let's get the show going. I will introduce our first student. We have Maxime Prieur, who is a master candidate in the School of Human Nutrition, and his talk is entitled, "Is Your Blood Sugar in Red?" Please.
Imagine that you have a very important call from your boss and that the 10% battery alarm shows up on your phone. You close up all your apps, the energy conservation mode kicks in, and you pray you can finish the conversation. Well, the same thing can happen to athletes. They train and train and train, and they can hit this low battery mode. And if they are not properly feeding for themselves over a long period of time, they can hit what we call relative energy deficiency in sports, or commonly named REDS. You can see REDS as the overly use your phone in this low battery state. It will just shut down. So the athlete will probably experience suppressed immune system, weakness in muscle, and even break their bone. And we know that this state of low battery mode is affecting 30 to 60% of our athletes. And it is really difficult to evaluate this problem in the field. So researchers are looking at glucose level in athletes. So we know from consensus guidelines that athletes are experiencing low sugar level. And researchers around the world suggest that we could use a small device inserted under the skin that will look at continuous glucose variation over 24/7. Named Continuous Glucose Monitor, or for the intimate, CGM. So my research will be looking at the use of this technology to evaluate this low battery state in athletes. But because this technology is commonly used for diabetes management, we'll also have to assess their acceptability of this technology in their daily life and training. So it is important to conduct this research because it will help shape the new evidence around the use of this technology for preventing athletes to be in low battery state. Make no mistake, the athlete's body cannot be replaced as the battery of your phone. So my goal with my research will be to identify to provide a small step into this field to identify how can we use this huge amount of data and if this technology could provide support for athletes to detecting this low battery state. And because the 15-year-old Max would be much happier flipping around than being in a cast. Thank you.
I need okay from the judges before I'm allowed to introduce the next candidate. Sorry. Thank you very much. Our next competitor is Denise Dutton, a master's student in Department of Natural Resource Sciences, and she's talking about what's a world without wheat. Thank you.
Imagine you're a farmer and you go out into your wheat fields one day in June to find that your crop, your livelihood, is barely hanging on in the hot, dry weather. This is what farmers had to deal with in Kansas last summer after a historic drought caused wheat yields to fall to their lowest level in 60 years. That a state that normally provides a quarter of the US's grain had to import it from Europe does not bode well for the future of this crop. And in fact, an analysis by the Woodwell Climate Research Center found that by 2050, the world could be facing a breadbasket failure every other year. Unfortunately, the current status quo practice in agriculture, monocropping, is not going to be resilient to climate change as we have seen in Kansas. So we face an imperative to redesign our cropping systems to be more resilient. But we don't have to reinvent the wheel to do this. Throughout the history of agriculture, diverse cropping systems have been used to increase yields and to act as a form of insurance against crop failure. When it comes to these systems, we currently know more about the benefits of temporal diversity than we know about spatial diversity, so growing different crops together in the same field. My research aims to understand whether diverse or unusual cropping systems, such as wheat and bean, might confer comparable benefits, and why. To do this, I'm looking at the microscopic level. So we know there are microbes that inhabit the cells of plants' roots and provide nutrients to their hosts in exchange for sugars. These microbes are effectively selected from the microbial community in the surrounding soil, and they can help maintain plants' health under harsh conditions. So by sequencing the DNA of the soil microbiome, I am looking at the diversity and function of these communities under a crop diversity and precipitation gradient. So if crop diversity is like insurance for farmers, then you can think of microbial diversity as for crops. My research will shed a light onto what extent the two are connected so that we can select cropping systems that cultivate resilient microbial communities and effectively safeguard the resilience and productivity of wheat. So why not cut out the middleman? Let's use the agricultural wisdom of the past to diversify our farms for the future, so we don't have to worry about a world without wheat. Thank you.
Our next competitor, Ekwa Manful. She's a master candidate in the School of Human Nutrition. Her talk is entitled Children's Vision, a Future Without Diabetes. Thank you.
I believe we've all heard of the song, Heal the World, Make It a Better Place. A powerful call to action that reminds us of our responsibility to build a healthier, brighter future for our children. Children with their innocence have complete dependence on their caregivers, and they deserve nothing less than our full commitment to their well-being. I have 3 children, and as a mother, I want what's best for them. My study is based in a community known as Kaniwage, an indigenous community located south of Montreal. 30 years ago, community elders such as Mrs. Emilia McGregor joined forces with researchers to launch a school diabetes prevention program to prevent type 2 diabetes, a type of diabetes that occurs due to certain eating habits or exercise habits. But why? About 50% of their children were either overweight or obese, putting them at a risk of type 2 diabetes. The impact of the program was positive, with the number of new cases of diabetes in the community stabilizing. And one will think that after 30 years of KSDPP's existence, the total number of people living with diabetes in the community will be low, but this is not the case. Diabetes remains a problem today. This means we need to continue with what was started. My research will go into the community and collaboratively reassess and redesign the KSDPP program using methods such as informal interviews, talking circles, and food mapping activities. In step 1, I'll re-engage with the stakeholders of the KSDPP, such as the school children, the school authorities, and the community. In step 2, I will identify the health and wellness priorities within each school. In step 3, I will assess what resources are available to ensure a sustainable plan within the school environment. Finally, I will recommend some recommendations that would include culturally relevant foods and exercise activities. If 30 years ago, people like Mrs. Emilia McGregor were able to start on this initiative, we can continue to build on this to ensure that in future, the children of Kaniwage can live healthier, happy lives. Some communities may even take inspiration and take action for their own children. There is hope. Together, we can heal the world, one community at a time. Thank you.
Our next competitor, Amanda Round. She's a master's student in the Department of Natural Resource Sciences, and her talk is entitled The Codfather, Decoding Survival. Thank you.
The Arctic is melting. And as it melts, species that have evolved to these freezing temperatures are facing a race against their own biology. You've heard of polar bears and belugas, but let me introduce you to the unsung heroes of the North, the Arctic cod. Arctic cod feed on plankton and in turn are eaten by everything else. Whales, seals, seabirds, even other fish species. They are the codfather of Arctic ecosystems. And without them, life in the North just wouldn't exist as we know it. Now, their bow ties don't help them survive, but their gill rakers, these small comb-like structures on the inside of their gills, do. It's actually how they feed. When young, their gill rakers are quite dense in order to capture small baby plankton from the water that flows over their gills. But as they get bigger and move to deeper waters, these gill rakers space out to capture larger, more mature plankton. But here's the problem. Climate change is pushing Arctic plankton out, and Arctic cod may need to adapt their gill rakers in order to survive on new or different prey. And that's where my research comes in. So, genes are like the instructions that tell your body what to do. I'm trying to identify the genes responsible for gill raker growth in Arctic cod to see if these genes and in turn their gill rakers can adapt. First, I will take genes known to instruct gill raker growth in other fish species, and then I will compare that to the entire Arctic cod genome to see if there are any matches. Once I have my matches, I will measure how much the gene is expressed in Arctic cod samples using a technique called quantitative polymerase chain reaction, also known as qPCR. Now, this sounds scary, but think back to when you were a teenager and you were in your awkward, pimply growth phase. Your growth genes were turned on way more than they are now as an adult. Your genes are always there, but it's a bit like a light dimmer. They can be turned up, down, on, or off. So, for example, if qPCR shows me that the growth gene in the gill rakers is turned on more brightly, this will mean that the gene expression is higher in that individual, and likely that fish's gill rakers are more dense. By comparing gene expression across age groups and habitats, this will help me determine when and if gill rakers are adaptable, which helps inform conservation strategies by allowing me to identify which factors make them most vulnerable to changing prey. But this isn't just about the conservation of one fish species, as Arctic cod are crucial for the Arctic ecosystem, which itself drives global nutrient patterns. So, if we lose Arctic cod, this would destabilize northern food webs, destabilize fisheries, harm Canadian economies and the communities, which are many, that rely on them. Factors which ultimately affect us all. Thank you.
Our next competitor, Preet Manchanda, PhD candidate in the Department of Plant Science. His talk is entitled, Searching for Bob the Builder of the Plant Leaf. Thank you.
Imagine the quiet aftermath of a cold winter. As nature wakes to the promise of spring, a remarkable transformation occurs in the barren trees. The dormant branches sprout lush green leaves. This rebirth of life gives rise to the most critical cellular layer of the leaf, the mesophyll layer. Sandwiched between the two boundary layers, the mesophyll layer is divided into two types. The palisade mesophyll, comprising of tightly packed columnar cells, acting like a solar panel to capture the sunlight, and the spongy mesophyll, which acts like a lungs for the leaf to facilitate the air exchange. Such a vital layer powers the process of photosynthesis, which produces the oxygen that sustains life on Earth. And thus, this layer needs a detailed study to uncover how it develops and identify who the Bob the Builder is constructing this layer. Unlike the TV character Bob, here we do not have a construction map, nor we have a team of rollers and mixers, but we have a genetic blueprint. This blueprint guides the activities of several genes in a coordinated manner. My research involves improvising on a molecular technique which helps me study these genes in a single time frame, and up to 50 genes can be studied simultaneously in one single leaf sample. I use tiny DNA fragments that can bind to these genes and emit vibrant colors such as blue, green, yellow. And I capture these lights, which actually represents the gene activity, through a confocal microscope. The microscope also captures the 2D images of the mesophyll. Then I create 3D models out of these layers and map those genes with different activities on those layers. When done across different stages of leaf growth, I can actually see in a real-time view as to how these layers develop and which genes are playing a vital role in their development. This research is extremely thrilling, as it gives a huge amount of data and opens the door for further research, such as gene editing can be used to enhance leaf efficiency in terms of photosynthesis and better crop yields. But at the end, nature is the best Bob the Builder, and we scientists have a lot to learn from it to craft a better future which can tackle pressing global issues such as food security and sustainable farming. This research will lay the foundation stone towards achieving those goals faster and efficiently. Thank you so much.
Our next competitor, Yi Jin, a PhD candidate in the School of Human Nutrition. Her talk is entitled, Moving Toward Better Health, Can Dairy Influence Diabetes Risk? Thank you.
Let's start with a quick poll. Raise your hand if you don't eat dairy like milk, yogurt, or cheese. Anyone? Raise your hand if you eat reduced fat dairy because you think the saturated fat is not healthy for the heart. Quite a few. And who here prefers regular fat dairy? Most of us. Okay. Saturated fat has given dairy a bad reputation. But the thing is, we don't eat nutrients like saturated fat alone. We eat whole foods. In fact, studies that tracked people's habits and health over a long time have showed that those who consumed more dairy, even regular fat dairies, do not have higher risk of heart diseases. Interestingly, we found that they have a lower risk of type 2 diabetes, a disease affecting nearly 4 million Canadians. However, these are observational studies, meaning that we can only say associations, but cannot say that it is the dairy that reduces diabetes risk. Many other factors, such as lifestyle, can also play a role. My research addresses this limitation through a controlled clinical trial where we directly test how dairy intake affects diabetes risk. So in this study, we recruited 60 middle-aged adults with pre-diabetes, meaning that their blood sugar level were above average, above normal, but lower than the diabetes cutoff. These individuals were randomly assigned to one of the three groups. So the first consumed no dairy, the second consumed 2 to 3 daily servings of reduced fat milk, yogurt, and cheese, and the third consumed 2 to 3 daily servings of regular fat milk and cheese. The intervention lasted for 3 months. During that, we asked participants to stay as usual, and we measured their weight, daily food intake, as well as physical activity. To assess their diabetes risk, we measured insulin sensitivity using the BEST test. So this tells us how well their body responds to insulin, a hormone that regulates blood sugar level. We also did a series of physical and blood tests to see their overall health. This includes blood pressure, blood sugar, inflammation, and blood cholesterol levels. We did all our tests twice, once before and once after the intervention, to see if the dairy really made any changes. So I'm currently analyzing my data to see if there are any differences among the 3 dairy groups. Once complete, my research would help clarify the role of dairy in diabetes prevention among high-risk population and hopefully inform dietary guidelines so that we can all move towards better health. Thank you.
Our next competitor is Caroline Hunziker, and she's a master's student in the School of Human Nutrition. Her talk is entitled, How Would You Like Your Loved One to Feel While Receiving Healthcare? Thank you.
I'll begin by asking you a question. How would you want your loved ones to feel while receiving healthcare? When I think of someone I deeply care about, like Mom, I want her to feel comfortable and respected. This is a picture of my mom after giving birth to me. Yes, I know, I look adorable. That smile on her face is something we all want for our loved ones, right? So what is my research about? Well, it's about what you're feeling right now. When you add a cultural component to it, it's called cultural safety. Specifically, I'll be looking at the dietetics program at McGill. Did I mention that I'm also a registered dietitian? I want all dietitians like me to provide culturally safe care to our patients, particularly Indigenous peoples. There are too many horror stories out there of Indigenous people facing discrimination while receiving healthcare, way more than people like me. This is deeply unfair, and we know it, and it's our responsibility to address it. Notice the shift in how you might be feeling right now compared to the beginning of the presentation. This discomfort you're feeling is exactly what my research is trying to address. My project is called Grounding McGill's Dietetics Program in Cultural Safety, a pathway to truth and reconciliation in Quebec. Its goal is to identify the facilitating and the hindering factors to incorporating cultural safety into our education. To do this, I will conduct qualitative research. For those of you here who don't like math, qualitative research doesn't deal with numbers. It's about people's stories and experiences. My first step will be to conduct a document analysis. I'll review McGill policies, course curriculum, syllabus, competencies, to conduct a baseline assessment as to how cultural safety is currently embedded within the program. Then I will conduct interviews, profs, faculty, and dietitians like me who work with Indigenous peoples to get their perspective on cultural safety. My last step will be maybe involving some of you. I will conduct a survey with students in the last years of the program, and then I'll conduct even a focus group, so a group discussion to clarify their answers. I will combine the findings from all of these steps into formal recommendations to improve our program. Ultimately, this project is about improving the dietetics program at McGill, hopefully inspiring other healthcare programs in this university and beyond to incorporate more cultural safety. It is our responsibility to improve the health of Indigenous peoples so that more people can feel like I would want my mom to feel. Thank you.
Our last contestant, Samantha Dizon, a master's student in natural resource sciences. Her talk entitled Beelining for Wild Pollinator Conservation on Cranberry Farms. Thank you.
Raise your hand if you love insects. I do. When some of you think of insects, you probably think of ugly, creepy crawlers or spreaders of disease, and some are. However, insects are the most underappreciated workers in the agricultural industry. Without millions of insect pollinators like native bees and flower flies, we would struggle to produce enough crop yield to feed our rapidly growing population. So we need to find ways on how to conserve and promote these wild pollinator populations because they're currently threatened by pesticide use and habitat destruction. And for the booming Quebec cranberry industry, little is known about how their agricultural practices are impacting these wild pollinators. So my research is focused on filling this research gap, specifically in the Centre to Quebec region, also known as the cranberry capital of the province. And by working directly with local cranberry farmers, I collected nearly 3,000 native bees and flower flies from their farms and then preserved them in 95% ethanol. Then back in the lab, I used a critical point drying machine to remove the ethanol, and it helps to maintain key characteristics to further analyze their taxonomic diversity and functional diversity. And in order to do that, I used dichotomous keys and field guides to identify the species of each pollinator. By measuring their species abundance and richness, it illustrates the taxonomic diversity of native bees and flower flies within the cranberry farms. Then I will be looking at their taxon, their functional diversity, specifically at 5 functional traits. Through existing literature, I will determine their habitat specialization and larval feeding preferences. This will provide insight into the behavioral characteristics that influences their ability to pollinate. Then I will use a digital microscope to quantify their hairiness, body size, and tongue length. These 3 functional traits will help me to understand how much pollen they can carry and distribute. So far, I have over 25 different species of flower flies and 1,200 specimens of flower flies. Once I've finished analyzing them, I'll continue and repeat this process with the remaining 1,800 bee specimens. By better understanding which pollinators are in these farms, I can tailor a guide for each cranberry farm based off their agricultural practices. So while insects may not be so cute or cuddly, they play an essential role in agriculture. So let's build a future where we provide these pollinators the best possible working conditions.
It's going to be a minute while he fixes my...
So our last competitor, Larson Jorgovic, a master's student in parasitology. His talk is entitled, You Can Hide, but You Can't Run, a tale of antigenic variation. Larson.
We've all had one of those sleepless nights. It's some days like this where you just can't fall asleep. Perhaps you're too excited to start a brand new job, or it's the fear of writing your exams tomorrow. Now, what happens when sleep remains outside of your control? Imagine you're an individual in rural sub-Saharan Africa, and surrounding you are the buzzes of blood-sucking tsetse flies, ones which land and siphon out your blood. However, this isn't just an innocent bite, as inside lies another parasite, which now has been injected straight into your bloodstream. This is a trypanosome. Now reproducing in your blood, it now travels to the center that defines us as human, the brain. Some time passes and you start having those sleepless nights. You are losing control of yourself, eventually leading to a state of comatose and death. As a scientist, we ask, how does the trypanosome win? Immune systems can clear most infections with ease. The trypanosome, however, has a trick up its sleeve. Antigenic variation, presenting to the world a beautiful coat, taunting our immune cells to come and attack. But right before they have a chance to strike, a new coat appears and the parasite hides in plain sight. My research is to better understand the complex protein interactions that make antigenic variation work. Now, protein in the context of molecular biology are pieces of a puzzle which come together to make biological systems function. The problem with my work, though, is we're missing a lot of those protein pieces, and we don't know exactly how they come together. So we're unable to paint that full picture. The good news is, though, with genetic engineering, we can track our proteins inside the cell, and we can have a better understanding of the antigenic variation proteins. Here, I can make those proteins glow bright green, so we can track where exactly they lie inside the parasite. From there, we can then use this tracker for something else. We can concentrate high numbers of our protein so we can use it as bait. We can then use it to trap, capture, and identify all other closely associating proteins. With this knowledge, we can create a map and discover previously undocumented protein interactions, clearing away some of those clouds and adding to our understanding of subsystems, which are key on the aisle of antigenic variation. One protein at a time, we will eventually have our map complete, which we can then follow to put an end to, which we can then put together to discover treatments to put an end to those sleepless nights. Thank you.
Give another big round of applause to all our competitors. I'm just going to skip the slides. So the judges are going to leave to deliberate. And during that time, we're going to have an open Q&A with the audience. So you'll get to ask our competitors questions about their research. You can ask questions about graduate studies. You can ask them questions about the 3MT competition. The floor is yours. I will launch the vote for People's Choice. So if you use the QR code and the short link on screen, you'll be able to vote for your favorite talk, and the competitor with the most votes will win People's Choice. The poll is going to be open until 5 o'clock, after which we're no longer going to be accepting votes. So during that time, the students are going to bring their chairs to the front and get ready for the Q&A.
You got a couple of minutes to stretch your legs, and then we'll start our Q&A. I'll open the floor to questions. Anybody has a first question? I can bring the mic over. Any particular questions? I have some here, so if you want, I can start. And I can ask, I'll ask Yi, how did you find the study participants?
Yeah, so for my study, it's a clinical trial. So we start to look for our participant at the hospital. So there were 2 endocrinologists on our team. So they helped to refer some of their patients, as well as some of their previous participants that were working with other projects. And then we created some pamphlets and put it around the community in the school, in the university as well. So, and after that, after we had maybe first 10, 20 participants, they actually had a great experience with us, so they referred to their families and friends. Yeah.
Great, thank you. Anybody from the audience will have the courage for a question, no? Yes.
Hello, a question for Ian as well. You're talking about the impact of dairy on diabetes, right? You talked about saturated fats, low-fat dairy, and regular dairy. Do you also consider the effect of antibodies in dairy products, especially milk? Perhaps there's an effect.
Yeah, thank you for your question. But for our particular project, we didn't account for that. And all the study products we provide to the participants were available from the supermarket. And this particular project, we really focus on the amount of dairy as a total, so milk, yogurt, and cheese, as well as the saturated fat content. But that's something interesting, and I think a lot of people are really concerned about, but it's not the focus of the subject. Thank you.
Thank you. Yes.
Thank you. I also have a question for Yi. Nice to meet you. So my question is just also based on because for myself, I have to drink the lactose-free milk. So did you make a comparable or consider about like reduce the lactose-free milk to 3.5% of the full like lactose-free milk for diabetes? Is this a fact or not a fact? Yeah.
Okay. Thank you for your question. So just a clarification, for this project, because we want to focus on dairy and dairy fat, we actually recruited only participants without any concerns of dairy. So for example, for you and as well for me, I'm lactose intolerant. So we cannot join the study. We cannot be part of it because we don't want too many noises in this study. This is why we kind of focus on people who can drink regular dairy products. But I think for your concern, let's say if we are comparing dairy products with varying fat content, but for lactose-free options, the fat content also differ. So I would say maybe the findings could extrapolate a little bit, but it would be hard to say. And there have been previous studies that look at people who are lactose intolerant and those who have no problem drinking milk, and they actually found those people have a lower risk of type 2 diabetes compared to those that who are not lactose intolerant. So that's definitely a field of interest in the future.
Thank you. I'll follow up with a question. I like math. So, question for Caroline and Ekwa. What issues do you face when working with qualitative data? Versus mathy. Mathy data.
Mathy data. I feel, honestly, I haven't yet entered the data collection phase, so it is hard for me to answer that based on experience. But I can answer, and that's also what makes it super exciting for me. Qualitative research is not necessarily something that I have explored as a dietitian per se, so I'm used to looking at very clinical data, clinical trials, and so it's getting that perspectives from the actual populations, the participants, their reasoning behind choosing certain things or behaving in a certain way that really, I figured out, interested me when I was practicing full-time. So I think it's a different type of challenge because I don't know about it.
Yeah, it's definitely a fascinating.
Exactly.
Yeah. Yes, sure. Would you like to add to this, Ekwa?
Yes. Also for me, I haven't gotten to the data collection yet or analysis, but what I can add is that it's very interesting because the feedback you get are diverse. And so when you're coding the feedback from qualitative data, you need to sort of do it with somebody from the community so that you can code rightly and then analyze properly. Else you'd use your own interpretation, which may not be the facts. So that's what makes it interesting really diverse feedback and you need, like support from somebody who understands the codes or the themes that come out from the community. Thank you.
Very interesting. Thank you. Anybody else? Go ahead.
Thank you. This is a question for Larsen. Could you explain me a little bit more about how the parasite cannot be attacked by our immune system?
Yeah, for sure. So this parasite, so I didn't say the species name of it, but this is Trypanosoma brucei, the African trypanosome. So the way that it works is essentially they present an antigen code and they do something known as VSG switching, variant surface glycoprotein switching. So the surface coats are these proteins, which are highly antigenic. And just as kind of I said in my little talk, they don't hide anything. They're alerting the immune system saying, hey, I'm here. However, because they can switch those variant surface glycoproteins, they have a bunch of molecular machinery, and that's kind of what my whole thesis is, trying to understand how antigenic variation works. So when those antigens are presented, they're being attacked by the immune system, then they just switch. The machinery changes, and then the transcription changes from one certain VSG being transcribed to,
Saved your life.
Yeah. to a different VSG. So it's kind of playing this game of cat and mouse. So the trypanosome's always under attack and the immune system's trying to get them. So they kind of hide that way. Yes.
I enjoyed your talk very much.
Thank you.
On the same note for the trypanosomes, how
Yes.
quickly do they change their antigen coding? Like how quickly is that happening?
Yeah, so that can vary. So basically, from my understanding, as I'm writing my thesis, so like an immune response can be established within 4 to 7 days from the adaptive immune system. So the switching is happening within that frequency. And actually what's really, really cool is not only one single VSG is presented, so one surface coat, in the population overall, you're actually having multiple different surface coats. But one over the course of time, like over those, say, like 4 to 7 days, becomes dominant. So then those immune systems get really focused on the one, and then they kind of forget the others, and then continues like that. Yeah, so about that time, yeah.
Yes, we'll go to the next question.
Thank you. This is also kind of a follow-up to those past two. How do you think you could translate your research into a treatment? Like once you understand the mechanisms of the protein, how do you think we could?
Yeah, for sure. So basically, there's a lot of understanding. So basically, it's really cool, first of all. So VSG switching is actually, I think, one of the most complex ways that a pathogen can do to avoid the immune system. So actually what's really interesting, I didn't mention it, but because the antigens are always changing, we're basically unable to develop what truly could be a vaccine. Because, vaccines present that antigen and then the immune system recognizes it, attack, bada bim, bada boom. But we can't do that because they're always changing, and how many different vaccines can we do? So we kind of scrapped that idea. So the way that I'm looking at it is, can we target the proteins that are allowing switching to happen? So if we're able to make a drug or some disruptor, a molecular disruptor that affects those proteins, then it's checkmate, and we got that parasite out. Yeah.
Thank you. Anybody else? Right, so I have a question for Samantha. So how can we help with wild bee population and saving or conserve the wild bee population in our day-to-day lives?
Well, first of all is our gardening and what we're actually planting. So I know a lot of people love those like really pretty ornamental exotic plants, but planting native flowers and incorporating that into your gardens could be really helpful for wild pollinators. Even dandelions, I know people think they're a weed and they're super ugly, but if you keep them in your lawns, they really are very helpful. There's also a lot of great resources online. I think there's like the North American Pollinator Campaign that has a lot of great resources that they devoted to conserving wild pollinators. They even have like seed packs that you can buy. And a lot of seeds also have that little indicator at the top corner of what pollinator they attract. So hummingbirds, butterflies, bees, those are great things to look for. Yeah.
So we'll all have dandelions next year in our lawns. That's it. It's official. Anybody else? Otherwise, Oh, yes.
I actually had a question about the coding you were talking about. You said you need someone from the communities to come in and, I don't remember, verify it. Could you expand on that? Because that sounds really interesting.
Okay, so what happens from my knowledge with getting qualitative data is that you're getting feedback from diverse people. And unlike quantitative where you have a formula and you do this, you add that, you might be saying something from your cultural perspective, I might be saying something. So assuming I don't come from your culture, I may not understand what you're trying to tell me. So that's what I meant. So you need to sort of like put the answers in themes and code them. And then you need to be able to explain what the meaning of the feedback you got is. So that's what I meant by the coding.
It's kind of like putting them in like cultural context. Yeah.
Yes.
Okay, cool.
Yes.
That's awesome. Thank you.
Very interesting. Anything else? So let me ask you, how did you choose your 3 empty slide? How did you come up with your slide? Anybody? You have a mic? We can pass it down.
Well, actually, I went through a couple drafts before I decided on this one because Ingrid told me it was the best one. But I wanted to show all my different insects that I collected. So those are photos that I took from a microscope, and they're my specimens. And I pretty much just edited it so I removed the background. And I wanted to showcase cranberries because it's the yield that's really the most important for the farmers at the end of the day. And it's the taxonomic and functional diversity that helps achieve that. So yeah, that's why.
Right.
So we all answer.
Sure, yes.
Perfect. So for me, it was important to relate the topic, Indigenous topic, to me, who I'm a non-Indigenous person. And I also wanted to show part of the reflective process that I am still going through while conducting this research as it relates and how to relate it to me or to experiences that I have lived, because I cannot get an Indigenous perspective from myself. And I just wanted to make it a little bit more personal and relatable to people who were non-Indigenous.
Thank you.
For me, the slides are the 3 groups of my intervention. So my intervention are 3 groups. And also I think it's a lot of people's choice. It reflects people's different choices of dairy. So that's why I chose that.
My idea for this 3MD slide was to demonstrate as what we do in the lab is 3D imaging and do the microscopy. So as a microscopy lab, I wanted to represent that. We do 3D imaging. We try to study how things develop in 3D as it is in nature, and how those things develop, which genes play a major role from what starts as a simple structures to grows out to be a complex structure. And that complex structure is sustaining life on Earth. So that was the idea, to represent the lab and represent work at the same time. Thank you.
Yeah, for me, so I had 3 points. So I know this parasite can be a little complex, so I wanted to put my focus solely on kind of making it make sense to other people. So I had the little diagram showing the switching process and how the parasite avoids. But then also I want to show, the work that I did. So actually that microscopy image is an image that I took under a confocal microscope. So that was kind of cool. And then how do you illustrate proteomics to people? So I thought of, we'll use a map. We're going to discover an island, and that's where I go. And X marks the spot. We can find that protein. It's right there. It's kind of cool. Yeah.
I mean, how many of you had heard about Go Rakers before my presentation? You guys, because I presented to you. But I wanted to make sure you could visualize what I was saying, because my work is a bit, we're studying this gene because of this physical change, because of this change that's happening in the ecosystem that's driven by another external thing. So I wanted to make sure you guys could see how important that change is and how important the Arctic cod were, which is why I called it the cod father and gave them little bow ties. Like Arctic cod, you probably, or have you heard about them before?
Yeah. Well.
Yes? Okay, I'm getting some yeses. But yeah, most people don't know how important they are, and I wanted my slide to kind of highlight that.
For me, my slide wasn't so nuanced or complex. I just wanted to contrast monocropping with intercropping, or just more diverse cropping systems in general. And so I chose the most atrocious example of a monoculture that I could find on the internet. And then for my diverse cropping treatment, I wanted to make sure it was still relevant to my study. So I found one of wheat and bean, not my plots specifically, but close enough. And I think it was pretty clear which one I was preferencing in the slide. So simplicity is better, in my opinion.
Thank you. So this wasn't my original slide, but for me, I wanted to reflect children in their elements. Children are happy. They're innocent, and that's what we want. We want them to live in a world where they don't have to suffer adulthood diseases. And so I just thought, let me just put happy children in a circle and also reflect the indigenous elders who came together to start the school diabetes prevention program. And Mrs. Emilia McGregor is one of the revered, elders in the community who joined forces with other researchers to start this program. And she was happy for me to use her picture, so that's how I came about.
Very nice.
So for me to choose my slides for this presentation, I wanted to be really simple in a way of talking about it. And since everybody, I believe, has a phone, it was easier for everyone to understand the simple concept. So I use Google because it has all the answers, and I found the image that was perfect for the presentation.
Perfect. I would like actually to follow up with a question for you.
Yes.
So the blood sugar monitoring that you have spoken about, can that be something used or beneficial for not only for professional athletes, but for people who also regularly exercise?
Yeah, so at the moment, there's no evidence that it could potentially give information about like health, performance, etc. So this is why we are doing research to understand a little bit more how can we use this tool to evaluate performance or even energy in my case. But there's this CGM that I talked about is commonly used for diabetes management, but it's expanding to other fields such as even like surgery or I'd have like other even like healthy people, but unfortunately there's not enough evidence, and the market is growing up, and we have to provide some evidence for the users to be able to use it properly.
Makes sense. Thank you. Yes.
I guess I just have like a general question for anybody who wants to answer, or it can go down the line, but what's something that kind of excites you in your field right now that could be like a new method that's come out or recent research or just something personal that you've reflected on that's kind of reignited your passion for your research? I know you're all at different stages, so I suppose like what in the beginning or at the end, but something that's really kind of you've reflected on.
Thank you.
I can start because I have the mic. For me, my research is really exciting because I have a background as an athlete, and now I'm doing research in like sport nutrition. And since I'm merging, my lab is focusing on type 1 diabetes mainly. So I'm merging two areas together using the CGM and the sport nutrition research. So for me, it's really exciting to have this kind of new angle for the field.
Okay, so I'm still at the proposal stage of my research, but because I have children, I just love children. And for me, type 2 diabetes, which is currently being suffered by children, is a thing that bothers me. And so I'm just excited because I can, whatever I'm able to come up with my study, it could be, inspired other communities to also take action. And who knows, I could go back home with the results and save more children. So that's about my study. Thank you.
Thank you.
Yeah, for me, I'm really excited about like the intersectionality of my subject and like how agriculture can be a medium to addressing so many different environmental problems. Like it all just kind of intersects, whether you're talking about like climate change mitigation or like enhancing biodiversity for its own sake, but also for like wild pollinators, like we learned. And then there's just such a human dimension as well with like of course providing food for the growing population, but providing autonomy for farmers and like local landowners, and just like allowing them to be in reciprocity with the land and like do what maybe they've always done for generations, and hopefully without such harmful consequences for the environment going forward, because it really doesn't have to be that way, and it could be so immeasurably beneficial as well. So I'm really excited about that potential.
Thank you.
It's not a method or anything, but potentially going to the Arctic. I actually just got a grant for that today, so I'm very excited for that. Thank you. Hopefully logistically that works out, but I'm most excited to go up there because up until this point my lab really hasn't collaborated with indigenous communities, and because it is so far north, most communities up there are mainly and like predominantly indigenous. So I'm really hopeful that I'll be able to work, because I'd be going up with the DFO, I'd be able to work with them to communicate with the communities. And they have so much knowledge that for so long was ignored, and now obviously that's getting a lot better. I definitely want to be a part of that and integrate their knowledge into my studies, or at least observations that I have in the field, and then communicate that back to those communities. That's already in place, but I do want to be a part of that.
So when are you leaving?
Oh, sorry?
When are you leaving?
Hopefully it would be like late August. It's in the Beaufort Sea, which is like above the Arctic Circle, so it's like cold all the time, and that sea is kind of treacherous. That's the only time I can go. But yeah, hopefully in less than a year.
Very exciting.
For me, so I'm wrapping up my master's, so I shouldn't be doing any more experiments. But as I'm wrapping it up, I'm analyzing a little more proteomics data, and I'm finding, pulling down that protein, looking at the other closely interacting proteins that are associating with some endonucleases, right? So with the whole VSG switching, there's a lot of RNA modifications and that can happen, and RNA modifications to allow it to work. And so, things that would be really cool to explore are what happens if we knock down this protein I pulled down? What happens, can we investigate more at the other interacting proteins? Do they form some sort of complex? How important are they for VSG switching? So yeah, getting kind of excited thinking about it, but yeah.
So you're staying on for your PhD?
Uh-oh. No, hopefully not. I'm just finishing up, yeah. No, I'm joking. I mean, it's really fun. I love doing it, but yeah, I'm ready for something else. Yeah.
Exactly.
The most interesting part of my project is that we have collaborated with 2 engineering labs. And so the idea is that the mesophyll layer comprises the major chunk of the leaf. And if you think a bit scientifically, is that the mesophyll is performing photosynthesis, and it also sustains all the environmental forces such as rain, snow, wind. And even after those environmental forces, the leaves are standing still on the trees. And so this ignites me and excites me a lot in understanding how the leaves are so smart in designing their structures that they perform the photosynthesis and they are also mechanically very stable. So it's kind of a trade-off that they have as it develops in nature. And this is what is very exciting to know. What nature is trying to build is very, very smart and very, very efficient. And the collaboration of engineering labs, learning those engineering kind of aspects with biology is really exciting about this project. So that's the idea.
My project is quite interesting for me because I'm also a clinical registered dietitian. So first, it attracts me because it's a clinical project where I still get the opportunity to work with people and deliver dietary suggestions and talk to people. And second, because the project really touched upon cardiometabolic health. And as we know, a lot of people experience diabetes, obesity, cardiovascular disease, and it's just so important. And lifestyle factors can really make a difference for these diseases. So I think it's really great value to put more study in it. And for people who are suffering from these diseases, they've already been told by a lot of doctors, healthcare professions to have a lot of restrictions or a lot of health changes. So for example, as the poll shows, a lot of people love regular fat dairy, but I guess a lot of people have also been told to stop eating regular fat and switch to lower fat. But there has been really a lot of evidence showing that there is no harm of regular fat. So I think my research really would add another layer of evidence in a clinical trial that we can show the actual cause and effect relationship between dairy and diabetes risk. And we also measure a lot of cardiometabolic factors so we can grasp an overall picture of the effect of dairy on health. So I'm actually analyzing my data. I have all my participants and samples collected. So I can't say now whether dairy is good or bad in terms of diabetes risk, but I think the results will come up in a year or so. Yeah, and we also had a lot of collaboration with endocrinology, with other university, and we are doing some lipidomics, proteomics, genomics data. So that's really exciting. I'm really excited to learn how to do those and to see if we can figure out the underlying mechanisms of how this happens. Yeah.
Thank you so much for your question. It's a very interesting one. For me personally, I think reconciliation is a word that has gained a lot more momentum recently. And as a dietitian too, I think food is such an amazing way to look at EDI, equity, diversity, and inclusion concepts, including reconciliation, because we all eat no matter who we are. And it's a great thing we have in common. The other thing that I would add as well, what excites me, qualitative research. There's a bloom in qualitative research recently, and quantitative is super important as well. But I just find it so fascinating to understand the human perspective and position towards the science that we put out in the world. What does it concretely mean for people to have that information? So this is why I do what I do.
Very interesting. Go ahead.
So when you have like 3,000 insects that you have to go ID, you really have to find the joy in the small things in doing that. So you're like, oh, thank God I found that one curled hair on their mesotibia. It means it's this genus and not that. So you really find the joy in small things like that, and it builds up, and you kind of build this skill. And you become really proud of yourself when you look at the whole and then you look at all the boxes. But one thing I love about my research is the community of entomologists. They're the weirdest, funnest people ever. They're like decked out in insect-themed clothes, and they will yap about a mite to your ear, and it's the best thing ever. It's amazing.
Thank you very much. Well, I see our judges are back. So I will ask, please, the competitors to pull their chairs to the side. And we'll have a fake drum roll. Thank you very much for all the questions, and thank you for answering. You have the mic? Okay.
You can give them the mic back.
I can give you the mic back. That's what the boss said. I can give you the mic back.
You're already snooping. All right, everyone. Thank you so much to the audience for voting for People's Choice. We truly appreciate your participation. Thank you to our wonderful judges for all your hard work. Dr. Alice Sharestis will now announce the winners for our 2024 competition.
All right, so we have People's Choice Award goes to Amanda Round. Amanda Round is from the lab of Professor Denis Roy. Congratulations. Here, please. Yes? You can continue. Okay, cool. Thank you. We have everything? All right. We have the third place winner. So the third place winner is Samantha Dizon. Please come up. Samantha is from Dr. Jessica Gillung's lab. Moving on to the second place. Second place winner is Preet Manchada. Please come up. Preet is from Dr. Anya Geitman's lab. And we do have our first place winner, who is Amanda Round. Thank you very much, everyone. Thank you very much.
You guys can go have your seat. I'm just asking the final question. Congratulations to all our winners and all our competitors. You guys did great. We would like to thank again Mrs. Lister and her family for their dedication and support. Your vision has thoroughly enhanced the science communication skills of our students and empowered them to share their research and their ideas, which we clearly saw today. We are very grateful for your impact on our students and our community, and we truly appreciate your partnership. A big thank you to our wonderful judges for participating. Thank you to Dr. Alice Sharestis. A big thank you to our wonderful multimedia team, without whom this event would not be possible. So Jonathan, Jamie, and John, thank you very much. Thank you, audience members in person and online, for attending. I wish you all a wonderful evening, and we hope to see you again next year. Take care.